is what you expect to see when you look at DNA, right? A double helix, a twisted ladder. What? Well, here's how I would draw it. You can do it. So, let me remove all those things. And on Friday, I told you guys that DNA is just a series of nucleotides, right? It's a chain of nucleotides with a complementary chain directly across from it. So what we're looking at here, this part of it, it's a lightsaber.
No way.
So here's our twisted double helix, right? Our ladder that we took and we turned. The sides of that ladder, are composed of sugars and phosphates all attached to each other. So on the sides, it's sugars and phosphates all attached to each other. And in the center, we have our nitrogen bases that meet. So, if we look here, this is one nucleotide. So it's one nucleotide. That's the second nucleotide. Third, fourth, fifth, and it just keeps going. That's 5 of 3 billion. And remember we said when we watched that movie that there were 6 billion total? Because then each of these 5 have 5 directly across from it. So here's 1, 2, 3, 4, 5, for a total of 10 nitrogen bases, for a total of 10 nucleotides.
You want us to draw that?
You should have, what I would make sure I had is the idea of a sugar and a phosphate. So yes, draw your sugar, your phosphate, and your nitrogen bases. And you should know that there are 4 different options here for nitrogen bases. The sugar here is deoxyribose. So the sugar is deoxyribose. That's where we get our name DNA from. D is for deoxyribonucleic acid. The phosphates here have a P inside the phosphate. So sugar, phosphate, nitrogen base. Now you'll notice, or should notice, guanine always pairs with cytosine. Cytosine, guanine, thymine, adenine. When we look at these, what's really important is that pairing. And the reason it's important is because if you think about it, it affects how wide our DNA is. If G always pairs with C, we always have 1, 2, 3 rings at the center of the ladder. If A always pairs with thymine, we have 1, 2, 3 rings across. So that's the width of the ladder. Let's just, if that happens in that instance, if I have, say, 2 thymines across from each other, so I have a T here and a T here, we call that a mutation. That particular kind of mutation is not uncommon in your skin cells. It's caused by UV radiation, and we call it a thymine dimer. Di meaning two, so it means two thymines. That's what causes skin cancer. So it's kind of a big deal when, just a little, it's kind of a big deal when the pairing is not correct. That's the kind of thing that causes skin cancer, something you're familiar with, and it's the same kind of thing that caused that little boy in the movie to have an autoimmune disorder. See how on this side it has a sugar here and then a phosphate? This is not a good anti-parallel image. Hold on. So the way that they're shaped here, what you can't see is there's a hydrogen hanging off on these spots and the necessary other chemical to hydrogen bond. This is able to make 3 hydrogen bonds where this can only make 2 because of its 3-dimensional shape. We group nitrogen bases into 2 different groups based on their size. The 2 different types of nitrogen bases are called purines and pyrimidines. So, purine the purines are those nitrogen bases that are 2 rings wide. So which of our structures are 2 rings wide?
The third one.
Which letter?
DNA.
2 rings wide. Here's 1. Here's 2. So adenine is a purine because it has 2 rings in its chemical structure. Is there another purine? Guanine is also a purine.
Now, is that the same for every example or just for this picture?
Every example. So guanine and adenine will always be 2 rings and they will always be purines. Our pyrimidines when do you test on this material, by the way? Next Tuesday. Because I told you guys last Friday. So you did remember it from last Friday. Your pyrimidines are thymine and cytosine, and they're only 1 ring across. God bless you. So if we were to straighten out our ladder and not make it twisted, you guys agree that's what a ladder looks like, right?
Yeah.
It has sides and rungs in the middle.
Well, it depends on what kind. Well, maybe there's some
What is
like
it,
that.
step ladder?
I have two.
A ladder where the rungs were different sizes wouldn't make much sense. So here they're always 3 across, right? If I were to mispair them, the DNA wouldn't have straight sides because the nitrogen bases would be either too large in the middle, they would be 4 rings across, or they'd be too small in the middle. They'd only be 2 rings across. So base pairing is absolutely essential. This is what it would look like if I were to take our twisted DNA ladder and straighten it out. This here is what we would call the sugar phosphate backbone. So this is the sugar phosphate backbone. And that is the sugar phosphate backbone next to it. So every DNA double helix has 2 sugar phosphate backbones on the sides. So the sides of the ladder are sugar phosphate. The rungs of the ladder are nitrogen bases. And they're nitrogen bases that are held together by hydrogen bonds in between them. So adenine and thymine hydrogen bond to each other, and guanine and cytosine hydrogen bond to each other. The way I always remembered, we call that base pairing, by the way, complementary. The way that I always remembered when I was in biology as a freshman, I remembered the pairing because adenine and thymine are made with all straight lines as letters. So they always go together. Cytosine and guanine are both made with the same initial shapes. So you'll always remember your pairs if you remember who looks alike. C and G look the same. A and T are all straight lines, look the same. And they're always going to hydrogen bond to each other. We call this anti-parallel. So DNA is called anti-parallel because they're parallel lines, right? They're next to each other. The anti part is because something else strange happens on a previous slide that you don't necessarily need to know, but. So here we have a sugar on the end, and here we have a phosphate on the end. It's because the backbones go in opposite directions. That's where the anti part of anti-parallel comes in. It's because they go in opposites. The number of hydrogen bonds that occur between our adenine and thymine and cytosine and guanine are different. Thymine and adenine are joined by 2 hydrogen bonds, and cytosine and guanine are joined by 3 hydrogen bonds. So these dashed lines are always hydrogen bonds because they're not real strong bonds in chemistry. They are temporary bonds that are easily broken. Now, how many of you are like, easily broken is a bad thing? This is my DNA. I would prefer it not be easily broken.
What if they do get broken?
They have to be broken. They have to be broken every time DNA replicates. So it is a very deliberate system. During the S phase of mitosis, DNA has to break all of those bonds to make an exact copy of itself. That's how you make new cells.
We will break all 3 billion
Mm-hmm.
at once.
It's just like a giant pair of scissors and then you go.
Actually, it's not one giant pair of scissors. It's multiple small pairs of scissors that operate all at the same time. That's your S phase of mitosis, and it has to happen very quickly and very, very accurately.
Why do the different line what? Like, why does GCF3 and ATM 2?
Because of the way that it replicates itself. The process of DNA replication is absolutely dependent upon this structure, upon the anti-parallel nature of DNA. And its complementary nature, so the fact that it fits together. The process of DNA replication is called semi-conservative. What that means when we say semi-conservative is every new strand, like every new double helix, is half new, half old. So it's half new, half old. It's because DNA is complementary, so that they fit together, A with T, C with G. That's how, that's the fundamental principle behind replication. And it occurs during the S phase of mitosis. So we're going to take a look at a video of DNA replication, and we'll watch it through, and then we'll watch it again so you guys can take some notes on what it looks like. These videos from the replication are already on PLUS Portals for the purposes of you studying for what day is that test again, Michael?
Tuesday.
Excellent.
Complementary to a small
What would you write first? And what attaches at the origin of replication? Helicase. What does it break in between the base pairs? Breaks the helix by dissolving the hydrogen bonds. And what has to attach to our open strands? Primase is going to attach a primer. So primase attaches a primer. And what enzyme comes in, our big pink Pac-Man enzyme? DNA polymerase attaches after the primer. So DNA polymerase after the primer and adds complementary base pairs. This continues continuously on the continuous strand, on the leading strand. Sometimes you may hear this referred to as 3 to 5 on the template. This is what we call the template strand. And 3 is, it reads from 3 to 5 on the template. We just name it that. It's a chemistry thing about the chemical shape of the backbone. For those of you who are absolutely dying to know, I could explain it to you later. On the lagging strand, primase attaches, DNA polymerase moves 3 to 5 on the template, but it does so in small fragments called Okazaki fragments. O-K-A Z-A-K-I. O-K-A Z-A-K-I. Okazaki. Eukaryotic cells have multiple origins of replication. So this happens many, many, many places all at once. Happens many, 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 many places all at once.